Plants, let's start with plants. Plants can be described as vascular or non-vascular. What does that actually mean though? Vascular plants are plants with a transport system for support and to allow different substances to be moved around the plant. So it ensures organs such as leaves, stems, roots, flowers, and seeds receive what they need, and it relies on xylem and phloem, right? Those two tissues that we were talking about for the transport of water, minerals, and gases. Non-vascular means it doesn't have a transport system for providing support or moving substances. It only relies on osmosis and diffusion to receive its nutrients and eliminate waste, such as mosses. Okay, so that's the difference between vascular and non-vascular. Vascular means it has a vascular system. Non-vascular, non-vascular. Non-vascular means it doesn't have a vascular system. All right, so like in animals, plant cells can become specialized, then they form tissues, then they form organs, then they form systems. So we have the root system and the shoot system. Okay, so the root system anchors the plant and absorbs water through the roots from the soil and inorganic nutrients as well from the soil through osmosis and diffusion. Its features to increase its surface area, it branches off into the ground, which aids with the anchorage of the plant. It has root hairs, which have a high surface area to volume ratio. It in fact increases surface area up to 12 times through extending from the root. And we have flattened epidermal cell, which have, of course, a high surface area to volume ratio. So we have diffusion for mineral ions and osmosis through the root system into the plant. If cell transport is too slow, facilitated diffusion or active transport may occur. Water moves through the epidermal cells that we're talking about in the epidermis, which is the most outer layer of the root. Okay, so that's a root system. The shoot system now, it usually develops above ground, okay, the shoot, it shoots up, and includes organs such as leaves, stems, and flowers. Its functions include photosynthesis, reproduction, storage, and transport. So it includes 3 different types of tissue as mentioned. So we have the dermal tissue, which is the outer layer of the leaf. It's responsible for gas exchange and waterproofing the plant. We have vascular tissue, which contains xylem and phloem tissues stored in vascular bundles. The xylem, as I said, responsible for water. Phloem, responsible for nutrients. It also provides structural support, and then we have ground tissue, which fills space around the vascular system in the stem. Okay, here is a diagram of the plant. So as I said, the root system, and then we start to get the shoot system. Okay, so we have the root cap into the root tip, these root hairs along the root, and then we get to the primary root, vascular tissue, and then into the stem, and we're shooting upwards to the shoot tip. Okay, yeah. Okay, so vascular tissue is the main transport system within the plant. As we know, it extends throughout the whole plant. The veins branch off the main vein, okay? Just like veins in humans, we have veins in the leaf, which basically carry nutrients the same way veins in humans do. They contain both xylem and phloem tissue, which aid in the distribution and transport of nutrients within the plant. Okay, leaf structure now. So photosynthesis is what we were just talking about, where it absorbs light, CO2, and water and converts it into glucose and oxygen. So it uses the glucose, but it'll secrete oxygen back out into the atmosphere, which is good for us because that means it's storing CO2 and releasing oxygen. But at night, when they don't have access to sunlight, ends during the day, but more passively, it will perform respiration, where it takes up some oxygen and releases H2O and CO2 to produce energy for itself. But its photosynthesis rate is much higher than its respiration rate. Okay, leaf structure. So it has an epidermis, which is on the top and bottom of the plant, and it prevents evaporation of water through creating a waterproof cuticle, and it protects the leaf. Okay, so the epidermis is just like our skin epidermis, protects us. It protects the leaf by creating that waterproof cuticle and kind of creating a barrier to the inside of the plant. It's transparent to allow light straight through it. The stomata is the primary gas exchange structure in plants, and it's in the lower epidermis, okay? The stomata has a guard cell which will open and close it. Just like how guards can open and close gates, the stomata has guard cells with its own gates, and the guard cells will decide when to open and close it. The guard cells, when they will kind of close involuntarily, the stomata, they'll close the stomata involuntarily. When the stomata fills up with water or gas, it will kind of force the guard cells to close, and then when it's all used up, the guard cells will open back up. Okay. Cells that carry out photosynthesis, we have two types, palisade cells and spongy mesophyll cells. Palisade cells contain a large number of chloroplasts, which we know are responsible for photosynthesis. They lie vertically and obtrude into the upper epidermis, aiding them to absorb sunlight, because obviously the epidermis is what's being exposed to sunlight. And then we have spongy mesophyll cells, which are secondary photosynthesis cells that are situated below the palisade cells and contain less chloroplasts, okay? Of course, palisade cells are the main ones doing photosynthesis because they have access to more sunlight. Okay, how does a leaf adapt to its different environments to allow it to continue to survive? Well, when there is a hot, arid, dry, barren environment, like in Central Australia, the leaves contain a waxy cuticle to protect against excessive evaporation of water, okay, because we have storage of water. And then rainforest environments where there's heaps of water, leaves are large and dark to allow them to absorb maximum sunlight in an environment usually lacking sunlight, because think a rainforest usually has a lot of trees covering other trees and other plants. And water loss is less of a concern, because rainforests will get continual rainfall. All right, so claims and conclusions, photosynthesis. So what is the difference in nutrient and gas requirements between autotrophs and heterotrophs? So the theory of photosynthesis, it's within the chloroplast of cells. Light energy is converted into chemical energy. And I've taken that straight from our biology course notes here at ATAR Notes. All right, this is mostly just the history of photosynthesis. I think, going to breeze over this one because, it's mostly the history stuff. It's really nice and easy. There's not too much to kind of learn about here. So the ancient Greeks hypothesized that plants were able to attain mass by absorbing minerals in the soil. Okay, they were able to grow from using the soil, which almost seems a bit obvious. In the 1600s, Jan Baptista van Helmont concluded that water led to plant growth, and giving plants sufficient amounts of water leads to better plant growth by conducting experiments. And Jan Eigenhaus, really hope I'm saying that German name, I think, correctly, was able to prove plants produced oxygen, okay? By noting that the plants placed in the sun produced air bubbles, and when they were moved into the shade, they ceased to produce air bubbles when they were submerged in water. Okay, because obviously oxygen and water, you can see the bubbles. If you've ever went under water and blown, bubbles form, and that proves that if a plant is producing those bubbles, obviously the plant is producing oxygen. Okay, in the 1800s, these next two names I'm really going to butcher, so I apologize if you're a native speaker of either of these languages that these surnames belong to. But Nicholas Theodore de Saussure, I hope I'm saying that right, showed that plants need both carbon dioxide and water to grow. Julius Sachs, I'm sorry if I again said that wrong, but they showed starch was produced by plants when light was present. And Cornelius van Niel proposed the general equation for photosynthesis, which led to our current and more simplified equation that we know, okay? Carbon dioxide, water makes oxygen and glucose, which we've went through today. Okay, so this was the original equation, and we've kind of really simplified it down into this. You do not need to know either of these two equations. This is just more the historical part of it. Really, you just need to know this. As I said, light is used in this process, but in cellular respiration, it's the opposite, except instead of light being used, we have just energy, ATP. Okay, so MRI, magnetic resonance imaging, which it uses radio waves and magnetic fields to generate an image of the plant. Okay, so this is imaging for plants and the xylem and phloem of plants. So we can use MRIs, we can use X-ray computed microtomography, and both of these kind of produce images of plants. So as I said, it uses radio waves and the magnetic field. When used in combination with technologies such as PET, so positron emission tomograph, we can also analyze cell function. The X-ray computed microtomography creates hundreds of images, and then it kind of combines all of them to generate a 3D image of the plant. It allows us to analyze the plant's internal tissues, just like a CT scan would, okay? So why am I telling you this? Because you could be asked to talk about either of these or both of these, or provide examples of imaging used to visualize the internal structures of plants. Okay, moving on. Function of structures in the plant. So what is the difference, again, in nutrients and gas requirements between autotrophs and heterotrophs? So the movement of the products of photosynthesis. So after we have this equation, and after we've produced our glucose and oxygen, oxygen we'll cover first. So the rate of synthesis is greater than the rate of respiration, as I mentioned. So excess oxygen is produced. It's basically released back into the atmosphere, as I said, which is good for us. We breathe in oxygen, and we don't want so much carbon dioxide, so it's good for us. Glucose, however, is what we're interested in. Glucose is transported by the phloem, which runs from the leaves to the rest of the plant, and it's transported by translocation. Okay, so it goes down from the veins, remember, down from the veins to the rest of the plant, depending on what part of the plant needs it. Okay, the pressure flow theory states that nutrients are moved into the plant by active transport. Why? Because it's moving, it could potentially be moving against the concentration gradient. Okay, remember that triangle, it moves from an area of high concentration to low concentration. If we have all of the nutrients coming down, we're going from an area of low concentration to high concentration, right? Because everything's coming down, everything's congregating downwards into the stem. So high concentration. In the leaves, there's a low concentration, low to high. So we need active transport to go against that concentration gradient. Water follows by osmosis, creating a pressure gradient downward. Nutrients move passively down phloem following the gradient, and sugars are transported out. Okay, gas exchange, microscopic structures. The diffusion of gases from an area of high concentration to an area of lower concentration, especially the exchange of oxygen and carbon dioxide between an organism and its environment. Okay, that is how we define gas exchange. Basically, diffusion except with gases. In plants, gas exchange takes place during photosynthesis. In animals, gases are exchanged during respiration, right, when we're breathing in and out. So, the stomata. The stomata is basically the number one way that gases leave a plant. So yeah, stomata, or stoma, which is singular, plural stomata. So it's located in the lower epidermis, as we saw. It consists of two specialized epidermal cells called guard cells, which surround a pore. So guard cells are on either end of the stoma or stomata, which either open or close to let out or let in gas. Okay, so during gas exchange, the guard cells swell to open the pore to release oxygen and gain carbon dioxide. When the stoma opens, water will also diffuse out. The amount of water which transpires depends on the climate and how much water the cell actually needs. So in a dry climate, the guard cells will not swell or will not become turgid, meaning opened, preventing gas exchange. Okay, so closed means it's flaccid, open is turgid. Okay, other parts of the plant use lenticels to exchange gas. Okay, so lenticels are found on the epidermis of different plant organs. So you 100% have seen this before on some woody plants. Such as fruit and the stems of woody plants. They are pores which always remain open, in contrast to stomata, which we know can open and close. Lenticels permit the exchange of gases between the environment and the internal tissue spaces of the organs. Okay, mammals, however. So mammals use respiration. So we use respiration in our lungs, specifically in our alveoli, to diffuse carbon dioxide out and get some oxygen in. So this happens through blood. So blood is the medium for transport within mammals. It carries nutrients and gases to cells and waste from cells. During the process of aerobic respiration, oxygen is transported into the blood in the lungs. Okay, oxygen diffuses into the blood. The process is conducted by the membranes of the alveoli, as I stated. The air sacs that are in your lungs, okay, they are called alveoli. So the oxygen from the outside world diffuses in the blood in your lungs, in the alveoli, and then the carbon dioxide leaves the blood in your lungs to the outside world when we breathe out. This is a close look of what alveoli are in your lungs, okay? They are contained in sacs, and they're basically air sacs, right? When you breathe in, that's where the gas congregates. Any of the many tiny air sacs of the lungs which allow for rapid gaseous exchange, airflow from the bronchioles of the lungs into the alveoli for gas exchange, blood capillaries abound around the alveoli which transport oxygen and carbon dioxide. When the thin membranes of the alveoli touch, gas begins to diffuse. Okay, So as you can see, when they touch the capillaries, either oxygen will diffuse in and then it'll go, this way to the heart, or blood needs to gain some oxygen, it'll go in, and then there'll be another one where it can kind of take out the carbon dioxide and, go around the body. Okay, so structural features of alveoli. So they have a large surface area, meaning they have a high surface area to volume ratio. This allows for easier and quicker diffusion of carbon dioxide out, oxygen in. They are covered in blood capillaries, so tiny blood vessels, which allow for high volume of blood for gas exchange. They are highly efficient because they're able to be the main site of gas exchange in the organism. And they're kept moist. Yes, they're kept moist. So carbon dioxide and water move quicker when dissolved. And alveoli walls are also very thin, only one cell thick, which allows oxygen and carbon dioxide to diffuse across quickly. Okay, continuing on to the next part of the syllabus, let's look at the lungs in general now that we've looked at the alveoli. So how do the lungs actually work? Well, air moves into the mouth from the external environment, okay, or the nose. It moves down the trachea, which is connected to the mouth. Eventually, the tube splits into two, one for each lung, okay? And actually, one will split into two more, and one just stays as one. Small branching structures are called bronchioles, which extend from the bronchi leading into alveolar ducts. The kind of passage keeps going down until we get to the alveoli. In the alveoli are in the alveolar ducts and they perform gas exchange for the organism. Deoxygenated blood is pumped into the lungs and then alveoli through the pulmonary vein. This blood becomes oxygenated and returns to the heart through the pulmonary artery. Okay, so the veins are always going away from the heart, arteries always going toward the heart. Okay. Other features, the diaphragm contracts and relaxes to let oxygen in and out, which allows us to breathe. The way we're able to breathe in a lot of oxygen is your diaphragm creates a vacuum, it creates suction and pulls in oxygen, air, not 100% oxygen, otherwise we would die, pulls it in. And then that vacuum suction releases, your diaphragm releases, it relaxes, and you can breathe out, okay? Just a little interesting kind of side note, in smokers, because they require so much kind of strength when they're breathing in cigarettes so frequently, their diaphragm starts to become overworked. Because think, how often when you're breathing are you going like really intensely and really intensely like smokers are when they're smoking? What actually happens is their diaphragm kind of, so diaphragms look like this under, so imagine the lungs are like this and the diaphragm's like this, and when you breathe in, does that, and then when you breathe out, it does that. What actually happens in smokers is because it's overworked and stretched, the diaphragm collapses. So it becomes harder for them to breathe in oxygen because there is no, it's more just, and then when they breathe out, whereas in a healthy person, it would be, okay. I know that was a bit of a random fact, but I think that's kind of interesting. And yeah, it is. I've seen the lungs of a smoker. It's very drastic, the difference between the lungs of a smoker compared to the lungs of a healthy person. You can definitely see the collapsed diaphragm. Okay. Yeah, so, okay, this is, I probably should have shown you guys the diaphragm image here instead of using my hands, but this is what I was talking about, right? You imagine as you breathe in, this diaphragm is sucking, it's going like this, as I was saying, whereas in a smoker, it's flattened, okay, because the overuse and the stretching. So it's like this, so it's harder to go. There's less kind of movement from here to here, from here to here. Okay. But this is kind of just an overall structure of the heart, just to kind of piggyback off what I was just talking about. So let's imagine you breathe in through your nose or your mouth. It goes down your trachea, which a lot of people call your windpipe, but it's your trachea. And then it goes into your left or right lung. Okay, I know that you're probably thinking, hold up, right lung. This is on the left-hand side. But imagine you're this person, right? You're facing us. It would be right and left. For us, we know it's left and right, but turn it around, right and left. So yeah, your right lung and your left lung. And then we have these bronchioles when it starts to split, okay? As I said, your right lung, your right bronchi will actually split once more before entering the lung, but nonetheless, that's not that significant. What you do need to know is that after we get to these bronchioles, we have our alveoli, okay? Yeah, here. We have all these alveolar sacs. They have a bunch of capillaries running all through them that will, depending on if they're deoxygenated blood or oxygenated blood, it'll either go to the heart or away from the heart, to the heart, pulmonary artery, away from the heart, pulmonary vein. Okay, so we do also need to look at fish. So fish have gills, which is the paired respiratory organ of fish and some amphibians, by which oxygen is extracted from water flowing over surfaces. So the gill arch, the gill arch holds gill filaments in place, okay? As you can see here, the gill filaments are kind of structurally bound to the gill arch. These gill filaments then branch out into the primary lamellae, which branches into the secondary lamellae, which we can see here, right? Covered in these similar kind of capillary structures, which will either bring in oxygen-rich blood and then oxygen-poor blood will leave with diffusion of oxygen throughout the body. Okay, so yeah, deoxygenated blood moves into the secondary lamellae in preparation for the water to flow in. So this would be when it's leaving, but water flow is coming this way. So as it gains, as it moves past it, it's gaining oxygen. And then water flows into the gill slits in the side of the fish. Oxygen is then absorbed from the water in the lamellae, which moves into the blood. Carbon dioxide is released. Like in humans, blood is then pumped back into the fish using the heart. The high surface area to volume ratio of these structures increases the rate of gas exchange. Okay, insects have a trachea as well, and as you can see, it's like almost identical to humans, same kind of structure going on there. So yeah, a large membranous tube which conveys oxygen from outside the insect to inside each of its cells. Okay. The difference between an open and closed respiratory system. So an open circulatory system is primarily found in invertebrates. Here, the blood flows freely through cavities, and there are no limited vessels to conduct the transport fluid. So kind of the entire body is saturated in blood. Whereas in closed circulatory system, which is found in vertebrates, the system has the presence of vessels that conduct blood throughout the body. So we have veins, arteries, capillaries. Okay, that so if you just cut into someone, you're not just going to have blood pouring out everywhere. The blood is contained in veins, arteries, capillaries. So open circulatory systems are common among small animals and insects. Hemolymphs and is pumped. Hemolymph is what we have in open circulatory system. That's what we call the blood, and it's pumped by the heart into a body cavity called a hemocoel, where it bathes the internal organs in nutrients and gases. It does not remain contained in vessels and instead has direct contact with organs and tissues. It's suitable for animals with low metabolisms that don't need quick energy, okay, like insects. So as you can see, the body cells are literally coated in the blood. Okay, the tracheae or trachea for single are, as I said, the circulatory and respiratory system of insects that allow for gas exchange. So in insects, air flows through small openings in the body called spiracles, okay, into the trachea and then into small branching structures called the tracheoles, just like how we had the brachia and the brachioles. Tracheoles branch into every cell in the body, allowing oxygen and water to diffuse in and carbon dioxide diffuse out. The movement of air occurs through passive diffusion or active ventilation, where larger insects contract their internal organs and abdomen to force air out. Okay, like when we breathe out, ventilation pushing the air out. So we don't have alveoli. We do have every cell in the insect's body will come into contact with this tracheole here. So it can literally just get delivered the oxygen directly to it. Okay, so common features between all these different gas exchange structures. They all have a large surface area to volume ratio. Why? Because it's harder to diffuse when there's a higher volume to surface area ratio. Because think, if there is let's say your school hall. Imagine your school hall. Actually, I know a lot of people actually don't have school hall, so I apologize if you don't have a school hall. Just imagine a school hall. Okay, school hall, right? Let's say I say to you, all right, you have to run into the middle. You run into the middle, okay? There's nothing there. It's an empty hall, whatever. You have to run into the middle. You're able to get there, let's say, in 20 seconds. Then I put big, I don't know, let's say big, I'm trying to think of something that's big and bulky. Let's say I put big bookshelves, or I line them all the way down the hall, all the way down the full hall. And I say, all right, now run to the middle. It's going to take you maybe like 20 seconds longer, okay? Because you'll have to kind of duck and weave between all these different bookshelves and kind of slip through the cracks and try to get your way through. It's the same idea here. When there's a lot of emptiness inside a cell, gas can, of course, get into the cell and get into the middle of the cell easier. When there's a lot of volume, though, when there's a lot of stuff inside, it's harder to get to the middle. Okay, that's why we want a large surface area to volume ratio, because they can get in quickly. We also want a moist, thin surface because it's easier to diffuse when it's thin and moist. Close proximity to a transport system that ensures that oxygen gets to cells and carbon dioxide is removed from cells. Okay, we want all of these structures have some sort of transport system that delivers the oxygen to all the cells of the body and removes the carbon dioxide from the body. It has a clear concentration gradient, i.e. gas on one side of the membrane is in higher concentration than the other. For example, oxygen is breathed in the lungs before it diffuses into the blood and travels to cells. Okay, digestion. What is the difference in nutrient and gas requirements between autotrophs and heterotrophs? So, Digestion is the process of breaking down food mechanically and enzymatically into simpler chemical compounds in the digestive tract. For heterotrophs, they must source all their nutrients from the external environment through eating or ingesting food, which can be broken into simpler nutrients. Okay, so us. Why is it important? Why is digestion important? Because it is the process through which we gain the nutrients from the food that we're eating. So blood can transport these nutrients to where they are needed in the body for energy or structural purposes. However, protein cannot be stored. That's why it's made by our cells, okay? There are two different types of digestion. There's physical or mechanical digestion, and then there's chemical digestion. So, physical digestion involves breaking food physically into smaller pieces, okay? Physically. Think of breaking food physically. So it starts in the mouth with the teeth, chewing, cutting, and breaking down the food. The food then moves to the stomach, where the churning motion continues to break it into smaller pieces. Your stomach literally is doing this to the food, which churns it. It's called peristalsis, okay? Or peristaltic motion or movement. This increases the efficiency of the enzymes that are involved in both the mouth to break down food and especially in the stomach. Okay, so chemical digestion, we're talking about enzymes here, okay? Enzymes are the main thing that are breaking down the food that we're eating. So yeah, involves the use of enzymes to chemically break down complex molecules in food into simpler forms. So for example, glucose from a complex carbohydrate and amino acids from protein. So complex carbohydrates are broken down into glucose, and amino acids, so this is a protein, we can break it down into individual amino acids. So essentially the complex molecules are broken down into their building blocks or smaller ones. So because the body can't take up whole proteins, however, we can take up individual amino acids. So organs in the digestive system. Let's start with the mouth. So the mouth, of course, is a first site of physical digestion. The chewing or tearing allows us to increase the surface area of food before enzymes act on them during chemical digestion, because, as we know, it's easier to tackle something if it has a higher surface area to volume ratio, just as we learned with the respiration system. Okay. Amylase, salivary amylase, starts to break down your food in your mouth, okay? So it begins to break down starch, which is a complex carbohydrate, into the simple sugar maltose. So I'm not sure if you've ever bitten into an apple and gotten like a release of like a weird taste in your mouth. Hopefully some of you guys know what I'm talking about and I don't just seem crazy, but if you've ever bitten into like especially a green apple and you felt a whole rush of something, that's amylase coming out, okay? And kind of like, yeah, being secreted to break down the apple. Chewed food is then shaped by the tongue into a clump called bolus and swallowed, okay? Which sounds a bit disgusting, but it is. It literally just becomes chewed up mash and is swallowed. Esophagus now. So bolus travels from the mouth into the esophagus through the process of peristalsis, which is what I was talking about, right? Those muscular contractions which push the bolus into the esophagus. It's, that movement. Your muscles literally do this to push it along. While it's moving, the breakdown of starch continues to occur. So there's still salivary amylase that's breaking it down. To ensure food doesn't move into the respiratory system, a flap of skin called the epiglottis blocks the respiratory system entrance. Yeah, so I don't know if you've ever accidentally had food like almost go down your trachea. It's not a fun experience, but typically when you are swallowing, your trachea is closed by an epiglottis, which doesn't allow food down there. Down your trachea into your lungs. Okay, stomach now. So the food moves from the esophagus through a muscle-ringed soft-walled tube, the circular sphincter muscles, which are at both the opening of the stomach and the kind of ending of the stomach. They both control the opening of the entry and exit of the stomach. Physical digestion continues here, so stomach churning, the relaxation and contraction of the stomach walls continues to break down food. But mainly how the stomach breaks down food is through its chemical processes. So we have gastric juices in our stomach, which contain water, hydrochloric acid, a lot of it, and the enzymes pepsinogen and pepsin, a lot of pepsin, combined with food to form a mix called chyme, which I'm sure you've probably heard before. So the acidic environment of the stomach, which is specifically between 2 to 3, more so towards that 2, causes the enzyme pepsinogen to be converted into pepsin. As I said, mostly pepsin. So what does pepsin do? Well, it breaks down proteins into peptides or amino acids, as I was saying, and DNA and RNA are broken into nucleotides, okay, which are the building blocks of DNA and RNA. I was going to say something. Yes, so that's why if you've ever had acid reflux, that acid is coming up from your stomach, okay, because as we know, the gastric juices are very acidic, so yeah, the acid's coming back up. Okay. Then if we continue down the digestive system, we have the small intestine, which comes first, right? So where was it? Sorry. Oh no, there was a diagram. I will find it. Wait, no, I think I did. Yes, sorry, here. As you can see, we go from the sphincter down into the duodenum, okay? Duodenum is the transitionary part from the stomach into the small intestine. So small intestine into the duodenum, and then into the jejunum and the ileum, okay? So duodenum, jejunum, ileum, and then we travel into the large intestine, which you can't really see it here, but it goes up and across, down, okay? Actually, no, sorry, up and across, down, that way, into the rectum. Okay, so it's made up of 3 regions, as I said, the duodenum, jejunum, and ileum. So chyme from the stomach enters the duodenum through the pyloric sphincter, which is a small muscular opening. It's controlled by our muscles, involuntary control, of course, because you don't actually think, all right, let me release the food. No, you don't do that with your stomach. It stimulates the release of pancreatic juices, which contain a mixture of the enzymes amylase, trypsin, and lipase, along with bicarbonate ions, which further break down the food. Okay, so glucose and amino acids are absorbed by the capillaries, whereas fatty acids and glycerol move into the lacteal. Water absorption occurs in the small intestine, as we saw there, right? Small intestine. Okay, how do the nutrients actually get to the cells? Well, digested food travels to the liver after being absorbed in the bloodstream. The liver is essentially for detoxifying the blood and ensuring that sugar, glycogen, and protein levels are maintained. It plays a key role in food metabolism. So the liver is really essential in this, and really essential in the regulation of nutrients and the concentration of nutrients in the blood. Oh yes, sorry, because I can already hear the comments, all the questions coming in about what the lacteal is. The lacteal is the lymphatic vessels of the small intestines, which absorb digested fats. Okay, so the remainder of undigested material from the small intestine then moves into the large intestine, as I said. Okay, this includes water, salts, and dietary fiber. Yes, especially that fiber. Not to get too graphic, but if you don't have a lot of fiber, it's going to be a very painful process. What comes next? I'll just say that. Vitamins A and K are produced by the large intestine and also get absorbed into the bloodstream. Okay, so the large intestine. So we have the sesame. You don't really need to know all of this. Probably all you need to know is the ascending colon, the transverse colon, and the descending colon. Okay, as you can literally see, ascending colon, transverse colon, descending colon. You don't really need to know sesame. I think that's some fissure. I can't even see lymphatic fissure. I'm not even sure what that says. Yeah, you don't really need to know all of that. Just kind of the main elements. So what does the large intestine do? Well, it performs the functions of converting food into feces and absorbing essential vitamins and minerals. vitamins, minerals, and water. Chyme enters the large intestine via the ileocecal sphincter and is mixed with beneficial bacteria that have colonized the large intestine and is then passed through the 4 regions of the colon by peristalsis. Again, that involuntary muscle contraction. Along here, you have a very particular microbiome, which refers to all the bacteria existing. So you have, of course, a bunch of bacteria in your digestive system in its entirety, but especially in the intestines. And of course, in a lot of developing countries where they don't have proper sewage, that's how a lot of people become sick with things like cholera, E. coli, because the bacteria that exist in the intestine are fine in the intestine, in fact beneficial in the intestine, but if once expelled, they're consumed or infiltrate the waterways of these developing nations, or they in some way, like crops become exposed to feces. A lot of the time it's chicken feces. Either way, it somehow is consumed by humans. It becomes really, really devastating and lethal in a lot of cases, especially in developing countries where you don't just have sewage containment as an issue, but you also have sanitation and education and practices and all that kind of stuff, which is really, really bad. We actually had a case not too long ago in, I'm not sure, but I think it was Central Australia, where a farmer actually had been digging near a river, and he actually dug into a sewage pipe, burst it, and wasn't able to contain it, and some of it expelled into the river. And we actually had an outbreak of norovirus, I believe, which is obviously a kind of virus, in the local community. I think around like 200 cases of norovirus just from contamination from feces. So while some bacteria and viruses are really, really good in your intestines, and we have a symbiotic relationship with them, which means that we can exist together, and we actually have a beneficial symbiotic relationship. So we benefit from our relationship with them living in us. They benefit with them living in us. While that's really good in the intestines, it can be really, really bad if it affects somewhere else in the body and is swallowed. And it's not just contained in the gastrointestinal tract. Okay, so while chyme moves through the large intestine, bacteria digest substances in chyme, and that's why I was saying that they're beneficial, because bacteria also help with digestion. Fermentation converts chyme to feces and releases vitamins. Gases such as carbon dioxide and methane are produced as a byproduct. Water, as well as ions and nutrients released by gut bacteria, are all absorbed by the large intestine and used for metabolism. Bacteria is what gives feces its smell, and what gives it its color is actually red blood cells. So red blood cells, when they die, they are yellow, and that explains the color of urine. But when you have heaps of red blood cells and heaps of red blood cells and heaps of red blood cells, you get more yellow and more yellow and more yellow, and eventually starts becoming brown, hence explaining the color of feces. Okay, so the elimination of waste, right? Large intestine is responsible for helping water and certain salts to absorb into the bloodstream and forming undigested material into a more compact solid substance, which can be expelled by the body. The remaining waste material, which is feces, is eliminated from the body through the rectum, then the anus through peristalsis. Okay,